Hello everyone, welcome back to our channel. This is Sans Lectures. If you've not yet liked, subscribed and followed, please do so. We still continue with our cell communication series. Previously we discussed the various forms of cell signaling. We said there is cell communication. We said there is cell signaling, cell adhesion and cell junction. We looked at cell signaling, we defined cell signaling, we stated its essence. We looked at the types of cell signaling, and we are looking at the receptors, which we are still continuing with. Last time we said there are two types of receptors: intracellular receptors and membrane-bound receptors. Then we looked at the first type of membrane-bound receptors, that was the G-coupled protein receptor. The G-coupled protein receptor, we said there are three different types: GS, GQ, and GI. Then we looked at GQ. Today we are going to look at GS. GS. If you've not yet watched those videos, please do. That information is very necessary. Then at GS, we said that the G-coupled protein receptor has three parts, three units. The alpha unit. This was our alpha unit. Alpha unit. The beta unit. And the gamma unit. So we said previously that when our signaling molecule binds to the receptor, which receptor is found on the cell surface membrane, it causes activation of this, what we call the cytosolic tail, or what other texts will call the transmembrane protein, which we said spans 7 times across the membrane, and it will cause another response here, whereby we say this alpha unit is bind with GDP, and we shall have a freely floating GTP. So what will happen is this GDP shall be replaced by GTP. So this GTP shall come here on the alpha unit and replace the GDP. So we said that this alpha unit is the one that will interact with other molecules. So we said previously the alpha in GQ that it interacts with phospholipase C, but under GS it will interact with adenyl cyclase. This adenyl cyclase, which is also another molecule found in the cell surface membrane. What happens when that happens? To interact with adenyl cyclase, this is our adenyl cyclase. This adenyl cyclase shall get ATP, which is adenosine triphosphate, and remove 2 phosphates. It will remove 2 phosphates. That means I'll be left with 1 phosphate. So this ATP shall be converted into CAMP, which is cyclic adenine monophosphate. Which is CAMP. So what happens is this, and our CAMP is usually the second messenger hormone. Our first messenger hormone was the signaling molecule on the cell surface membrane. So CAMP is usually our second messenger hormone. CAMP shall then interact with a kinase C, which kinase C in this case has 2 parts. Number 1, it has the regulatory unit. Then number 2, it has the catalytic unit. So CAMP will always bind with the regulatory unit. It will come here and bind, and it will cause the two to dissociate. So I will end up with 2 things like this. This one has my CAMP. My CAMP shall bind to regulatory unit. So when they dissociate, this catalytic unit shall be free to go phosphorylate other molecules in order to trigger an intensified cellular response. I repeat, we said when a signaling molecule binds on the cell surface membrane, it triggers a transduction pathway. In this case, our transduction pathway, GDP is replaced by GTP, then the alpha unit shall interact with adenyl cyclase. Adenyl cyclase shall then convert ATP into cyclic adenine monophosphate, which is CAMP. Then CAMP shall bind to the regulator unit of protein kinase C. Then the catalytic unit shall interact with other proteins to trigger a cellular response. Another type of the G protein coupled receptor is GI. GI is the G coupled protein receptor inhibitory. It has a similar pathway. Only that when the signaling molecule binds here, it shall prevent the alpha unit from activating adenyl cyclase, and we shall have no response. Thank you very much.